myosin ceases to be in contact with actin filaments as a result of. Okay, so myosin, this is your myosin, this is your actin. So when ATP comes, it is dissociated. It is a very, I mean to say, often repeated concept that when ATP comes, it dissociates head of myosin from actin. So myosin ceases to be in contact when attachment of ATP happen to the myosin head. But let me discuss this. So myosin ceases to be in contact as a result of attachment of ADP to myosin head. Okay, correct. Sequence. Okay. See, sliding filament theory is very important. Every time I discuss this topic, every time I will revise the same thing. Why? Because in the exam we forget. The only way you can get a good marks, number one is your emotions, limbic cortex. You must be very much emotional about your study. And number two is your revision. Short-term memory will convert into long-term memory by long-term potentiation in the hippocampus. So every time you are revising the same concept again and again, it will become your long-term memory. So your emotion and your revision strategy is very important, doctor. So at rest, myosin heads are bound to ADP. At the resting condition, they are not bonding to the ADP, and are said to be in the cogged position in relation to the thin filament which does not have calcium. So until calcium is present, okay, thin filament is made up of what? Thin filament is made up of actin, troponin and tropomyosin. Thick filament is made up of myosin alone, and they are cogged position. You are cogged. What does it mean? That although actin and myosin they are together, but who is preventing tropomyosin? Tropomyosin cover what? Myosin binding sites on the actin filament. So at a resting condition, they will look together like a happy couple, but they are not. They look happily married or happily together ever after, but they are not, because tropomyosin is preventing the sangam of actin and myosin. So there cannot be contraction. Following an action potential, when calcium is increased, now this free calcium will bind to troponin C, and it will cause conformational change in the thin filament and allows myosin head to cross bridge with the thin filament. When troponin C is attached by calcium, it causes conformational change and uncovers myosin binding site on actin filament. So now actin myosin will attach, and now it's called as cross bridge. So first it is cogged position. Cogged position, number one, when calcium is not there. When calcium is there, it is called as cross bridge. Cross bridge will be formed between actin and myosin in the presence of calcium. Now head of myosin will rotate. After that, they are attached. Now head of myosin will rotate, move the attached actin and shorten the fiber. It is called as power stroke. So number third stage, when myosin head will rotate and Newton's third law, opposite and equal reaction, will be power stroke. They will bring two sarcomere together, two Z line will be together, two Z line will be closer, and sarcomere will shorten. It is called as your contraction. It is because of power stroke. At the end of power stroke, ATP binds to new exposed site and cause a detachment from actin filament. After power stroke, ATP will come and detach head of myosin. So head of myosin detachment is by ATP. That's why relaxation is a passive process. And after that, ATP is hydrolyzed into ADP and inorganic phosphate, and again recog. So we start from cogged position, then cross bridge, then power stroke, and then detachment of head of myosin by ATP. Please revise this sliding filament theory. It's very important for your exam.